At 4,290 meters, a camera found a ghost. 10 years later, science still cannot give it a name. In 2016, the remotely operated vehicle Deep Discoverer was crossing a basalt outcrop northeast of Necker Island in the Hawaiian archipelago. Then the seafloor seemed to move. Resting beneath the vehicle's lights was a small octopus. Its mantle was only a few centimeters long. Its body was pale, soft, and almost translucent. It had no fins, no dramatic color, and none of the heavy armor people imagine an animal would need at this depth. The camera was 4,290 meters below the surface. Most octopuses recorded at extreme depth belong to a finned group often called Dumbo octopuses. This animal was different. It was an incirrate octopod, an octopus without the fins and finger-like cirri associated with those deep-sea relatives. At the time, no incirrate octopod had been documented this deep. Scientists watching the dive could see that the animal was unusual. They could not place it comfortably inside any known description. It was almost certainly undescribed, and it might not even belong to a genus science had already named. People following the live expedition gave it a nickname before taxonomy could give it anything else. They called it Casper, but its depth was only the first mystery. Casper did not look built for the abyss. It looked unfinished. Most familiar octopuses can transform their appearance using chromatophores, pigment-filled organs that expand and contract beneath the skin. Color can help with camouflage, signaling, hunting, and defense. In the first Casper footage, those pigments were barely visible. The animal appeared white to nearly transparent, with internal shadows showing through its soft body. That ghost-like appearance created the nickname. It also created an easy explanation. Perhaps 4 kilometers of darkness had simply erased the need for color. But science has not confirmed that explanation. The reason for Casper's striking lack of pigment remains unknown. The camera shows what the animal looks like. It does not reveal the evolutionary path that produced it. And the body is not as helpless as it appears. There is no large gas-filled chamber inside an octopus that must resist being crushed like a sealed container. Its tissues exist at the pressure around them. The animal is delicate, but it is not a shallow water body accidentally surviving in the deep. It belongs there. The more difficult question is where, exactly, it belongs in the tree of life. A nickname can be created in seconds. A scientific name needs evidence. A video can record color, posture, movement, and the visible arrangement of the arms. It cannot reliably reveal every structure taxonomists use to separate one octopus from another. It cannot provide DNA. It cannot preserve the beak, reproductive organs, internal anatomy, or microscopic details of the suckers. For a formal description, scientists normally need a physical voucher specimen that other researchers can examine again. The specimen becomes a permanent reference, evidence that can be measured, compared, challenged, and reinterpreted decades later. No Casper specimen has ever been collected for scientific study. That leaves researchers in an unusual position. They can watch the animal walk, they can measure its depth, they can compare its shape with known groups, they can say it is probably undescribed. But they cannot confidently decide where its branch begins. A camera can prove that an animal exists. It cannot always prove what that animal is. There is also a quiet tension inside the solution. Collecting a specimen could unlock its anatomy and genetics, but the animal appears rarely, and its population size is unknown. A future expedition would have to weigh the scientific value of collection against the cost of removing one of the few individuals ever seen. Until then, Casper remains famous without being formally named. and it did not vanish after 2016. For years, the original encounter looked like a biological accident, one pale body in an ocean too large to search. Then similar octopods appeared again. In 2023, an expedition in Papahanaumokuakea Marine National Monument recorded a Casper-like octopus walking across the seafloor more than 2,300 meters down. In 2025, another was seen in the waters of the Cook Islands at a depth greater than 4.1 kilometers. These observations extended the story, but they did not solve it. Without specimens or genetic samples, similar appearance does not prove that every sighting represents the same species. The name Casper may now describe a visual type, small, pale, finless deep-sea octopods that resemble the original animal. Some may be the same species, some may not. Each sighting makes the animal more real. None makes its identity certain. The gap between popularity and knowledge has only widened. By 2025, the original Casper had become the most favorited animal in a game where players help classify deep-sea imagery. The public had adopted it completely. Science still lacked a specimen. The strongest clue to how animals like Casper may survive did not come from the original individual. It came from two mothers guarding eggs in the Peru Basin. The mothers were not hiding inside caves. They were suspended beneath the dead stalks of sponges. A 2016 study examined camera surveys from manganese-rich regions of the Pacific. Across the Hawaiian ridges and the Peru Basin, researchers recorded 29 incirrate octopods representing two distinct species. The brooding animals in the Peru Basin were not proven to be the same species as the original Casper. That distinction matters. They cannot be used as direct evidence of Casper's confirmed breeding behavior. But they revealed what reproduction at this depth can require. Each female guarded a clutch of roughly 30 unusually large eggs. The eggs were about 2 to nearly 3 centimeters long, and each one was attached separately to the stalk of a dead sponge. The water was close to 1.5 degrees Celsius. Cold slows development. Researchers concluded that the embryos, and therefore the mother's brooding period, could take years. For an octopus, brooding is not a brief visit. The female guards the eggs until they hatch. She keeps water moving across them and protects them from sediment and predators. In shallow species, that commitment can already be extreme. In the abyss, low temperature may stretch it across a significant portion of the mother's life. The image is easy to misunderstand. The octopus is not simply resting beside a plant. The dead stalk above her is the structure holding her future off the mud. The eggs depend on the sponge, and the sponge depends on a stone. Much of the abyssal plain is soft sediment. For kilometers, there may be almost nothing solid rising above it. Polymetallic nodules change that landscape. These dark, rounded rocks sit on the sediment surface and grow through the slow accumulation of iron and manganese oxides. Over millions of years, they can also concentrate nickel, copper, cobalt, and other metals. To a sponge larva, the value of a nodule is not chemical. It is physical. The nodule is an anchor. A stalked sponge can attach to it and lift its body into the water. After the sponge dies, the stalk may remain. That stalk can become a place where a deep-sea octopus fixes her eggs above the seafloor. The chain is simple. No hard nodule, no anchored sponge. No sponge stalk, no elevated nursery. Researchers had already seen what happened after nodules were experimentally removed in part of the Peru Basin. The community of stalked organisms that relied on them had nearly collapsed in the disturbed area. This does not prove that the original Casper depends on manganese nodules. It does show that some closely related, still unnamed deep-sea octopods can depend on a habitat structure that looks, from a distance, like nothing more than scattered stones. In the deep ocean, a rock can be part of an animal's life cycle. And these particular rocks are valuable to another species as well. Azu. For an octopus, a nodule can support a nursery. For humans, it can be an ore deposit. Polymetallic nodules contain manganese, nickel, copper, cobalt, and other elements used across modern industry, from batteries and electronics to specialized equipment and infrastructure. Their abundance on some abyssal plains has made them targets for mineral exploration and proposed commercial recovery. The demand is real, so is the uncertainty. A collecting machine would not remove only metal. It would remove the hard surface itself, disturb the surrounding sediment, and alter a habitat that develops at a geological pace. The most honest scientific statement is not that mining has been proven to erase Casper. It has not. The honest statement is that the biological cost is still incompletely measured. Some effects are direct. Animals and attached organisms in the collector's path would be displaced or removed. Others could extend beyond the track through sediment plumes, noise, changes in the seafloor, and the loss of structures that cannot quickly regrow. A nodule may take millions of years to form. A sponge community may not return on the timescale of a human project. An octopus mother cannot replace an egg anchor with a policy promise. This is why the Casper story is larger than one charismatic animal. It exposes a timing problem. Technology can identify a resource faster than biology can describe the lives built around it. Exploration is moving forward, while many deep-sea species remain known only from brief video encounters. The danger is not that science has already counted everything that could be lost. The danger is that it has not. 10 years of sightings have given us an outline. A small incirrate octopus can live more than 4 kilometers below the Pacific. It has little visible pigment. It walks across rock and sediment. Similar animals occur in more than one region. Related deep-sea octopods may brood large eggs for years on sponge stalks attached to mineral nodules. But the central questions remain open. We do not know Casper's scientific name. We do not know whether every pale sighting belongs to the same species. We do not know its full distribution, its diet, its lifespan, its population size, or why its body carries so little color. We do not know whether the original species uses nodules for reproduction, or whether that dependency belongs only to its deep-sea relatives. The next decisive discovery may be a carefully collected specimen. It may be a non-destructive genetic sample. It may be repeated observation of the same population, or a mother found with eggs in a place where every part of the habitat can be documented. Deep-sea exploration rarely gives science a complete animal story. It gives seconds of light, a location, a depth, and a chance to ask a better question. Casper is not unknown because nobody looked. It is unknown because the ocean reveals itself one narrow beam at a time. We found this animal before we understood it. Whether its world remains unchanged long enough for science to catch up is now part of the mystery. 10 years later, the ghost is still real and still unnamed.